Okay, we'll just open the session. We're gonna give like a couple of minutes for everyone to join us, and we will be ready to go in a second. There's still some people joining from the waiting room. So yes, just 1 or 2 minutes for everyone to be joining, and we will be starting this webinar in a minute. Okay, so yeah, I think that I'm gonna just launch it and we will be starting. So good afternoon, everyone, and welcome to this webinar. We are just receiving more people coming from the waiting room and hope to have everyone joining in a few seconds. So welcome to this webinar entitled KASP Markers Linked to Key Agronomic Traits for Chickpea Breeding. This activity is organized in the framework of the Horizon Europe Project BELIS, breeding European legumes for increased sustainability. My name is Joaquin Balduque. I'm an agronomist and coordinator of the plant production, health and breeding area within the training unit of SIAM Zaragoza in Spain. SIAM Zaragoza is the Mediterranean Agronomic Institute of Zaragoza, the Spanish Institute of SIAM, which is the International Center for Advanced Agronomic Mediterranean Studies. We are leading the work package dedicated to the BELIS Breeding Actors platform, networking, communication, dissemination, and transfer. So, and before we start this webinar, and for your information, we have enabled the question and answer option for the audience in this Zoom session. So as you can normally see in here, you will find, if you look closely to this image, you will find the option at the bottom of the part of the application you are using for joining us in Zoom. So if you have any questions from the audience, please type them there and we will address them at the most appropriate time. So I would like to thank all the attendees today who are joining us, as well as all the BELIS colleagues who will be presenting during this webinar. And just to finish with this opening, the main objective of this session today is to address the use of KASP markers for marker-assisted selection in chickpea breeding, as you may know, as well as the status of the breeding and seed production in southern Spain. Also, the stakeholder network being established within BELIS Project will be introduced, including its goals, activities, benefits of joining, and the steps to become a member. So after this opening, I will stop here, and then we will move to our first presentation. As outlined in the program, we are going with our BELIS Project Coordinator, Dr. Bernadette Julier from INRAE, France, who is going to present the project BELIS. So, Bernadette, thank you very much for being there, and the floor is yours.
Yes, thank you. Good afternoon to everybody. Can you see my screen?
Yes.
Is it okay? Okay, good. So I am the project coordinator of this of BELIS. This is the second webinar held online, and thanks Joaquin for organizing this webinar. Thanks to all the participants for participating to this meeting. I hope you will appreciate this webinar, and you will be invited to join the next webinar afterwards. I would like also to thank all the speakers today for their contribution to this webinar. So I am scientist at INRAE, the French National Institute of Research for Agriculture, Food and Environment, and located in Lusignan, in the mid-center west of France. And I am a geneticist working on alfalfa, mostly in Lucerne. So just to have a very short presentation of myself. So what is the concept of the project? It starts with different steps going from legume breeding to the use of these varieties by the farmers. So we start from the legume breeding in breeding companies or public institutes that release some improved varieties, and these varieties go to registration process. And if the varieties are registered, they go to the market, and there is exchange with evaluation and recommendation to market varieties. And all is done with in the framework of different types of cooperative models. So this is we are working within these different steps of activities around the legume breeding. And of course, we have identified the number of lock-ins at the genetic level, not enough genetic progress. Some lock-ins at the regulatory level, including registration process. Some lock-ins at the recommendation level with not enough information to the farmers, and also some financial lock-ins with reduced financial capacities for legume breeding. So we are addressing these lock-ins mostly a lot with genetic at the genetic level to better exploit genetic resources, to propose new breeding methodologies with better phenotyping and low-cost genotyping, and to go to breeding proofs of concept of our ideas for more efficient breeding for legumes. For the regulatory and recommendation lock-ins, we are also trying to improve registration process and recommendations. And about the financial lock-ins, we are studying different case studies, 24 case studies, and we have 4 living labs to improve and describe and improve how to breed budgets around legume breeding. And finally, we are also establishing a BELIS platform composed by a network of all people who are interested by genetic progress of legumes. So breeders, of course, extension services, registration offices, researchers, and also the industries. And we have also a portfolio of training and dissemination with online material, but also videos, training session, workshop, webinars, and this webinar is part of this activity. Yes, this is the overview of the project. Right. So the 3 main objectives of BELIS are first to develop tools and methodologies for cost-effective breeding programs, and also to deliver proofs of concept with and for breeders. We want to facilitate the economy and regulatory environment, so for variety registration, variety recommendation, and cooperative models. And we want also to implement an efficient, ambitious, and durable transfer of innovation through the BELIS platform that includes a network of breeders and all actors from scientific research, extension services, seed, food, and feed industries, as well as a training portfolio. So we are working on 14 legume species, 7 grain legumes and 7 forage legumes. You have the list here, going from the most cultivated ones to the least cultivated ones, but all of them are important. Grain legumes are mostly used, I would say, for food, human food. Humans mostly eat grain legumes. It's very seldom that they eat forage legume. These 2 types of legumes are used for feed, but they don't feed the same animals. Grain legumes are mostly used for monogastrics, while the forage legumes are used to feed ruminants. And all these legumes provide ecosystem services, and probably that forage legumes provide more services because they fix more nitrogen, but all of them are important for the agriculture. The partnership, so yeah, just to come back to the legume species, we are not working with the same intensity on all species, but we are working on all species. The partnership is composed by different types of participants based around the breeders. The breeders are at the heart of the project. We have research institutes and some of them are also breeders, they are underlined in yellow. We have one registration office. We have an education partner here, and we have, of course, seed industry represented by breeders, and also a cooperative of a breeder cooperative. And we have also 3 partners who are who belong to extension services. And this flower has leaves or leaflets on other stakeholders, consumers, feed and food industry, and farmers. Even if we are not working directly with these actors within the project. And you can see on the map the countries that have at least one participant in BELIS project. This project is, in the European vocabulary, is an innovation and action project. So at the end of the project, we will reach a TRL, so technology readiness level 5 to 7. So we are going to the demonstration of our ideas for breeding. So it's done with and for breeders, but also other stakeholders. A few examples of expected results to invite you to join the next webinars and to read our produces. Sorry. For example, we are working on high throughput phenotyping. For example, here to estimate plant height and biomass from drone flights, or more exactly from images taken after drone flights. And we would like that partners implement this technology on their plots with their material, and they applicate this result on their breeding in their predict programs. But about phenotyping, we are also working on the refinement or sharing other phenotyping methods related to disease resistance, quality traits, insect resistance, or drought resistance on some of the traits. We are also developing high throughput genotyping, and one key result that is upcoming is the delivery of a grain legume genotyping array. So it's a multi-species array. It's done to reduce the genotyping cost below the cost of a plot. And but we have also other work dealing with genotyping, and we are genotyping plant collections, including genetic resources, but also breeding material for better use of genetic diversity. And we want to show how markers could be used in distinctness test in variety registration in the future. We are developing tools for molecular breeding as a way to speed up genetic gain. So developing markers associated to traits, but also to develop prediction equations for genomic selection. And this will go up to proofs of concept in breeding schemes. And about the improvement of variety registration, we are testing a system in which we are using the phenotyping capacity in the European countries, and we are testing the relevance of a EU network at an agri-zone scale. So instead of considering each member state, we are considering across the member states some regions that have similar climates, for example, a Mediterranean climate or a Nordic climate, or yes, different types of climates. And so we are testing this network with trials in different countries using the same common control varieties, and we sum all the data, and the idea would be that each member state take its decision about the registration or non-registration of candidate varieties, but the trials are established on different countries. We are also trying to improve variety recommendation. So far, we are gathering data from variety values and market, and also to model the adaptation of species and variety type in the EU. And we also have to improve breeding cooperation among actors of the breeding, and to better support legume breeding. So we are studying some case studies and living labs to see what are the cooperation between partners. So between private breeders, public breeders, research, but also cooperative extension services, and maybe processing industries, and to see how to support legume breeding. We are also analyzing the effect of public policies on the capacity for breeders to continue legume breeding. So, of course, one objective is also to communicate and disseminate results and information during the project and beyond. So we have established the BELIS network to activate flow of information and cooperation among actors. So I hope you already belong to this network, but you will have more information by the end of this webinar. And the final aim of this project, among others, of course, is really to increase nitrogen fixation and protein production in Europe for sustainability and sovereignty. And of course, you know that, of course, nitrogen is very important, and recent events show that it's really very important. So thank you for your attention, and I think we can continue with the other part of the webinar. Thank you.
Thank you very much, Bernadette. Thank you for this presentation of this project. We are moving to the next slot in the agenda, which is with Dr. Ignacio Solís from the Spanish company Agrovegetal, which is a BELIS partner, whose lecture is entitled Chickpea Breeding and Seed Production in Southern Spain. Ignacio, thank you very much for being there, and the floor is yours.
Okay, good afternoon, everyone. I will participate in this talk like the spokesman of a group of people from different institutions working in the south of Spain in chickpea breeding and also in seeds production. I start with this nice slide with plant of chickpea and also very big and white seeds of the traditional cultivar Blanco Lechoso. During the last third of the 20th century and the beginning of the 21st, the chickpea breeding in Spain has been done mainly by public institutes, some of national institutions like INEA or CSIC, and also regional institutions like IFAPA, Itacil, SIA, ITAP, IMIDRA, and also University of Córdoba. In some cases, there is really breeding, in other cases, is selection of cultivars from ecotypes. From this work, some commercial cultivars have been developed, mainly of winter types, and mainly with small or intermediate sized seeds, like Badil, Fardón, Cuaíz, Castuvo, Amelia, Eulalia, but not so much, a small amount of certified seeds, because more than 95% of the chickpea seeds have been farm-safe seeds or not certified seeds delivered by the packers to the farmers. At the beginning of the 20th century, the most sought cultivars were Badil, Amelia, Eulalia, the main part breeded by public institution, but developed by commercial seeds companies. There is also one Italian cultivar, Vulcano, from Proseme. The main part of them, the certified seeds was of winter types, small or intermediate size. Probably the most growed cultivars were big size seeds, but not certified, reused from year to year. I will talk about Agrovegetal because we started the interest in chickpea at the beginning of 20th century, 21st century. In 1999, we started the production of chickpea certified seeds from cultivars of one company called Coipesol Semillas that nowadays is part of the group Syngenta. And this company, in collaboration with the University of Córdoba and IFAPA, they developed the varieties by Bianca, Crema, Baghdad, and Cairo. And one of them, the cultivar crema, was a successful variety because he has very good attitude for commercialization, pre-cooked in jars. There is a company interested in the harvest of this variety, and we produce certified seeds, and we made contracts to produce this cultivar. At the same time, Agrovegetal requests to the public institution in INEA the conservation of the very old public cultivar of the 80s, Blanco Lechoso, with very high cooking quality. Is the cultivar in the picture. But both crema and Blanco Lechoso are very susceptible to Fusarium and Ascochyta rabies. And then the chickpea area was very limited due to these constraints. And the main part of the chickpea consumed in Spain comes from Mexico and Canada. In 2002, Agrovegetal decided to start a chickpea breeding program in collaboration with the cooperative Campo de Tejada to develop spring types with big and white grains and with genetic resistance to fungal diseases. Why we started this program? Because the program at this moment at the University of Córdoba and IFAPA were not interested in this kind of big and white seeds. Then we started alone, but very, very fast we have the help of the University of Córdoba and IFAPA because we have signed an agreement to receive the help of the public institution with markers, crosses, and so on. I'm going to explain later. We started like a private collaboration, but very soon we make one thing that I'm going to explain later. 10 years later, we started this work. The cultivars Ituzi and Italica were released and now are probably the more successful cultivars in the seeds market in Spain. This is the figures of the chickpea area in Spain, not also in Andalusia, the whole Spain. When we have the first cultivar we had in Agrovegetal, Ituzi, arrived to the market in 2012. And at this moment, the area was around 20,000 hectares. At this moment, we are around 60, 70,000 hectares. The production is around 1 ton per hectare. These are the figures of the chickpea in Spain. In this market, these are the figures of the chickpea seed production from 2014 to 2025. When we arrived to the market, the total production in Spain was 158 tons of 35 seeds. 10 years later, is 10 times more, 1,465 tons of 35 seeds. You see that the seeds production is very clearly growing. There is some peaks. In 2018, we arrived to 900 tons. Later, we decrease. It's typical of our Mediterranean production that the yield is very regular and also the seed consumption is very irregular. Depending on the previous year, people buy seeds or not. But it's very clear that we have multiplied by 10 the certified seeds used in Spain. There is some cultivars like Badil from a public institute in Extremadura, but commercialized by Semillas Valle, that is in the market long time ago, but the ceiling is around 200 tons. It's one of the most successful. Italica is growing now, but the most successful cultivar is Ituzi. It's arrived to the market at the beginning of the figures, and it was growing, and the most important amount has been in the last season, 2025. Also, there is a cultivar called Casin, is growing also, and the cultivar Ituzi, it represents alone 1/3 of all the certified seeds in Spain. It's a type Blanco Lechoso, spring type with very white and big seeds. Okay, the cultivars produced by Agrovegetal are in green, and it represents 57% of the Spanish market. Then it's very clear that the role of Agrovegetal is very important in the certified seed of this crop in Spain. These are the figures of the last season with 1,466 tons, and the more growed cultivar was Ituzi, the second one Badil, Badje, the third one Italica, Casin, and so on. From the point of view of the ratio of certified seeds usage, in 2018, the certified seed usage ratio was around 13%. Now, with a growing market, we are around 18% of certified seed. It means that this amount of certified seed is making interesting this crop for Agrovegetal and for other companies. This is the schema of one of the BELIS case study is the production of the traditional chickpea cultivar blanco lechoso. It's not cultivar blanco lechoso and the derivatives like Ituzi, because Ituzi is a derivative line from this population blanco lechoso. This case study is working 20 years ago. At the beginning of the chain are the University of Cordoba and IFAPA, making basic research, applied research, creation of new genetic variation. Later, Agrovegetal is involved in testing and selecting and variety registration, and also the maintenance and breeding of the new cultivars. And finally, the cooperative Campo de Tejada is involved in seed multiplication, sales of seeds, and also producing chickpeas and marketing the harvest. This is one of the case studies in BELIS. And also, we have a living lab, one of the 4 living labs, where we want to increase the genetic base of germplants needed for different purposes to validate markers of quality and disease resistance needed for fast and effective selection of some characters, and to coordinate the different type of substitution for one common goal. The actors are the same, IFAPA, University of Cordoba, Agrovegetal, Campo de Tejada, but we want to open our target not only for blanco lechoso types, but also for other types like Castilian, Castellano, that are intermediate size, Pedrosillano, small size, and also chickpea for green consumption in salads, also chickpea for hummus, and chickpea for feed for animals. Very important interactions among farmers, seed company, university, public institute for agricultural research, and regional and international levels. This is one slide of each one of the actors of this collaboration. At the beginning of the chain, the IFAPA, a public institution for agricultural research in Andalusia. The main stations are in Cordoba, but they have a lot of training and research centers, 16. More than 1,000 hectares of experimental fields. They have germ plants banks, molecular and chemical laboratories, a lot of facilities. The second actor is the University of Cordoba, one of the most important public universities in Andalusia. And from the point of view of agricultural and food technology, is one of the best in Spain. Around 20,000 students, more than 1,000 teachers, and so on. They have degrees program for food and agriculture and science and technology, and also humanities and health sciences. It's one of the most important agricultural research institution in Spain. And following the chain, we are the seed company Agrovegetal. We are a small company, but we have 12 partners, and some of the partners are big partners. Like, for example, Cobab is the most important company producing milk in Spain, a cooperative. Also, we have the most important industry for pasta, but we need a lot of partners because we work only in autogamous crops, in wheat, triticale, chickpea, peas, and then we need a lot of collaboration in crops where to sell seeds is not very profitable. You know that the autogamous crops, the farmers can choose between to buy seeds or to reuse, and then you must buy very, very cheap if you want to sell. This is the reason you need to produce a very huge amount of seeds to be profitable. Here you have the 9 places where we can do field trials. For chickpea, we have every year trials in Escacena, Écija, Jerez, and Conil. For wheat, we have more locations. One of the partners is Cooperativa Campo de Tejada. Campo de Tejada, we are going to look later that they have one protected geographical indication, Escacena chickpea. They are traditional chickpea producers. Normally, it's like a dry product, but they want also to invest in pre-cooked in jars. These are the farmers of the company. These are the manager and also breeder. And Inmaculada Izquierdo is the person who consecrate more time to chickpea breeding in this cooperative. And where the selection and breeding program is 50% Agrovegetal, 50% Campo de Tejada. This is the protected geographical indication to produce Escacena chickpea by Campo de Tejada and also several other producers of the region. Then, this is the present, and for the future, we are creating a living lab, developing a living lab to have new types of chickpeas and to create a chickpea breeding cluster for developing new cultivars adapted to all the needs of Spanish farmers and consumers. In our living lab, in the chronogram, we have 3 main activities. The first one is to validate markers of disease resistance and also flowering time. This is the subject for the next talk that we will have later with Patricia and Natalia. The second activity is to evaluate jointly with farmers in field trials a set of 20 new advanced lines of chickpea of different types in 2 locations during 2 or 3 seasons, 2 years, sorry. And the third activity is to coordinate better the institutions that are working for a common goal. It is scheduled 2 meetings. The first one was done at Escacena last year, June 2025, and the second meeting will be at Écija in 2027 with representatives of University of Córdoba, IFAPA, Agrovegetal, Campo de Tejada, and others partners. This is some pictures about the first activity. Here we are taking samples of the leaves to validate markers. There's Natalia and the people from University of Córdoba. Also Jose Rubio from IFAPA. This is one important activity. We are going to talk about it later. The second activity is part of the seed selection program in Agrovegetal in collaboration with Campo de Tejada, is to evaluate in field conditions new varieties, type Blanco Lechoso, also type Castellano, also type Pedrosillano, in several environments, several years, to look for new cultivars, commercial cultivars. And the third activities are the meetings, the workshops. And the first workshops was the 4th of June, 2025. And in this workshop, we visit all the trials in the experimental farm. Later, we have 4 hours of different speeches. And here were the speakers. The first one, I'm going to see one slide about each subject. That was the first talk made by Jose Rubio and Patricia Castro about the validation of markers. Later, the second talk was about the breeding, the practical breeding in field conditions and variety development. It was made by Inmaculada Izquierdo of the cooperative Campo de Tejada and also Clara Marquez from Agrovegetal. Here you have some pictures to show how are the Blanco Lechoso types, the small types, and all the segregation. At the beginning, we only work looking for Blanco Lechoso type. We only wanted the big and white seeds, and the rest were discarded. Now we change the mentality. we are trying to have cultivars of different sizes and colors and destinations. The next step is the seed multiplication and production of certified seeds, where are involved the people from Agrovegetal. Here you have some pictures about the production of chickpea seeds. The first generation is made by hand, later machine. We have the machinery for select the seeds. And here is the plant, one of the plants of Agrovegetal selecting garbanzo, that is chickpea. The next step is the cultivation in the field of the farmers. Two farmers, Jaime Rodriguez and José Ortega, from the cooperative Campo de Tejada. They talk about the problems they found when we grow chickpea in Spain. The main problems are diseases, Fusarium and Ascochyta, we are trying to resolve, but also orobanche, problem with weeds, problem with cycles, with drought, and so on. The next step of the chain was the trade. And then the managers of three cooperatives, Campo de Tejada, Cooperativa de Conil, and Cooperativa Coesagro, they explain how they trade and commercialize the chickpea productions. Normally is very well traced, farmer by farmer. There is a sack for each one to have the price for each quality, because the quality is very important for the price in Spain. A very good quality, you can receive more than €1 kilograms. Intermediate quality, 80 centimes. Bad quality is 50, 60 centimes. It's very important the size and the color to have more price in Spain. Later, we have a speech from Pedro Benitez. He's the manager of a private company called Legumbres Pedro. This company, Legumbres Pedro, in the province of Cádiz, they sell dry seeds, but also pre-cooked jars and other products more I don't know the word. Not only directly the seeds, but also cooked seeds. And the last talk was made by Romualdo Ortega, that is a seed producer and also a chef that with a lot of expertise cooking different types of chickpeas. He talked about the geographical protected indication Garbanzo de Escasena, and to finish the meeting, he prepared 3 different plates: a salad with chickpeas, this plate called potaje, and the third one was a dessert made with chocolate and chickpeas. This was the workshop of the Living Lab, and the last news I want to give you is that we are asking for a new project, a new operational group called Garbantech, where we are going to put together the geographical protected indication of Campo de Tejada, Escacena, with another one that is in Córdoba. In the last year, the farmers and chickpea producers of Córdoba get a new European protected geographical indication, Garbanzo de Córdoba, Garbanzo de la Campiña de Córdoba, chickpea from Córdoba, and the 2 geographical protected indication together, we are going to invest to have more knowledge about new cultivars and also creativity in the cooking of this species in Andalusia. And it's all. Thank you very much.
Thank you very much, Ignacio. Thank you very much for this presentation. I would like to remind the people participating in this webinar that we have enabled the question and answer section in Zoom. So if you have questions, we will have some minutes at the end of this session for questions and answers. So please don't hesitate to put your questions in the Q&A section of the Zoom. So Ignacio, thank you very much. We are now turning to speak to one of today's main topic of this session, which is also giving the title to this session. This section is as mentioned, the KASP marker linked to the key agronomic traits for chickpea breeding by Dr. Patricia Castro from University of Córdoba and IFAPA and Dr. Natalia Gutiérrez from IFAPA, both members of BELIS Consortium. Patricia, if I'm not mistaken, we are starting with you. So thank you very much for being there, and the floor is yours.
Okay, thank you very much. First of all, I would like to thank the organizer for giving us the opportunity to participate in this webinar and to share the progress we have made so far in chickpea research. It is a pleasure to be here and to contribute to this exchange of knowledge. So to give you some context about where the results come from, our research group brings together scientists from IFAPA and from the University of Córdoba, and we have been working together on chickpea breeding for more than 20 years. So this long-term collaboration has allowed us to integrate different areas of expertise and to develop a strong and consistent research program. Although my colleague Natalia and I are the ones presenting today, the results we will show are the outcome of a joint effort, and they reflect the dedication and contribution of all the members of both groups over the years. So over this time, we have developed new chickpea materials, performed genetics and molecular studies for disease resistance, adaptative traits, and quality traits, and develop molecular markers linked to them. In this talk, we will focus on 4 main traits: Ascochyta blight resistance, flowering time, Fusarium resistance, and leaf type. However, within Project BELIS, our efforts have been centered on 3 of them: Ascochyta blight resistance, flowering time, and Fusarium resistance. So having defined the main traits we are focusing on, the next aspect is the plant material that makes this work possible. The development of reliable molecular markers strongly depends on the ability of well-characterized material and segregating population. In this context, our breeding program has generated a valuable collection of plant material. In our breeding program, we have developed 15 chickpea varieties and 80 real populations from different parental lines, and these populations segregate for important agronomic traits and are essential for building genetic maps, conducting QTL analysis, and identifying markers linked to the traits of interest. These populations have been a key plant material for the BELIS project, since we use them to identify SNPs linked to the traits and to develop KASP marker. So what is our role within the BELIS project? Our contribution has mainly focused on work package 4 and 5, which are the work package our group is involved in. And in this talk, we will explain how we have developed KASP marker for key traits such as flowering time, Ascochyta blight resistance, and Fusarium resistance, and how we are using the markers in work package 5 for marker-assisted selection. And as you know, developing effective molecular markers for breeding is not straightforward. It is not something that can be fully achieved in a 4-year project. It requires many more years of work. But before presenting the result we have obtained so far, I would like to hand it over to my colleague Natalia, who will explain the methodology we use to develop the KASP markers.
Thank you, Patricia. Can you hear me?
Yes. Yes.
Good afternoon, everyone. My name is Natalia Gutiérrez, research at IFAPA. I'm going to talk about the methodology to develop a KASP marker assay. KASP stands for Competitive Allele Specific PCR. It's a powerful and high precision marker technique used to detect genetic variation in individuals. Specifically, it targets single nucleotide polymorphisms known as SNP, as well as insertion and deletion at a specific loci. Because KASP markers are biallelic and dominant and codominant marker, they allow us to clearly distinguish between homozygote and heterozygous genotype. So how does it work? The method relies on allele-specific primer and fluorescence-based detection. Each allele generates a distinct fluorescent signal, enable accuracy in genotyping. KASP markers are fast, sensitive, reliable, and cost-effective, making them ideal for large studies and plant breeding applications. In particular, they are utilized used in market assisted selection, helping to enhance and accelerate conventional breeding programs. So, how do we develop a KASP assay? In the following slide, I will describe how to develop a genotyping assay using KASP marker step by step. The step 1 is the most critical and challenging step of the process because it is identify the right SNP or indel that are linked to the agronomic trait you are interested in. For example, Patricia, in chickpea, a lot of research has focused on traits related to disease resistance, especially against Ascochyta and Fusarium, which are two fungal pathogens that reduce the yield in chickpea. Also, important adaptive traits such as flowering time and leaf time have been studied in chickpea. The identification of SNPs or indel associated with this trait required extensive genomic and genetic study, but once those markers are identified, they can be used to develop reliable KASP assay. The second step is to obtain the flanking sequence around the SNP marker. In this example, I am showing a deletion that is associated with leaf type. Plants with a simple leaf have one base deletion, while the rest of the sequence is the same both phenotype. To design the assay, it's necessary to retrieve about 75 base pair ups and downstream of the SNP. Later, my colleague Patricia will go into more detail about this work. The third step is design locus-specific KASP assay by the company LGC Genomics. Now, the KASP reaction have two main components. First, the KASP assay mix. This is the part that's specific to your SNP or indel of interest. It's included 3 primers, 1 common primer, and 2 allele-specific primer. Each allele-specific primer has a different tail. One is linked to the fan fluorophore and the other to the X. These tails are what allow us to distinguish between the two alleles during detection. The second component is the KASP master mix. This is the universal component, meaning it can be used with any KASP assay mix. It contains 2 fluorescent reporter acid along with the all standard PCR component, including Taq polymerase. In this case, KASP uses a specially modified version of Taq polymerase, which improves the specificity of the reaction. So once everything is designing, the final step is to validate the KASP assay in our lab. For this, we need a real-time PCR system, the KASP assay mix, the master mix, and of course, the DNA sample. An important point here is that we already need to know the phenotype of the sample we are testing because they show differ for the polymorphins we are targeting. This allows us to check whether the genotyping result match the expected trait. We also need to genotype a sufficiently large number of individuals to properly evaluate how accuracy and robust the KASP marker is. And finally, we calculate the efficiency of the assay by comparison the genotyping data with the known phenotype across the set of individual used. And once the set is validated, it becomes a reliable tool that can be used in a breeding program. For the last, here I show you a schematic representation of an allele discrimination plot from KASP genotyping. Samples that are homozygous for allele E, labeled with FAN, show as blue dot, and the cluster is close to the y-axis. On the other hand, sample homozygous for allele B, labeled with HES and show a red dot, the cluster along the x-axis. Heterozygous sample, which carry both alleles E and B, produce both FAN and X-SINA at similar intensity. As a result, they appear in the middle of the plot and show a green dot. Finally, the black square representing the non-template control, which should not show any fluorescence signal. This clear clustering is exactly what we're looking for. It shows that the assay is robust and that we can confidently distinguish between genotypes. With this, I conclude my explication, and Patricia will continue with the result and show how this marker has been application in chickpea.
Okay, so thank you, Natalia. How did we develop a KASP marker for flowering time? To do this, we first analyzed 4 real populations, segregating for this trait. In this table, we can see the parent that we use for the crossing, together with the phenotype of each of the parent. These populations were evaluated under sort day condition, and then they were genotyped using GBS. So from the association analysis, we identified several genomic regions that are specific to certain population. For example, here the region in chromosome 1 that was only in population 12, and the region in chromosome 4 that is located in population 1, here highlighted in yellow. Okay. Also, there are some other regions associated with flowering time in multiple populations. For instance, in chromosome 3, highlighted in green, that is present in population 1, 8, 10, and 12, and also the region in chromosome 5, highlighted in pink, that is present in population 10, 8, and 12. So for developing the KASP markers, we focus on the region on chromosome 3 because it is associated with flowering time in multiple populations, suggesting that it is stable and relevant across different genetic backgrounds. So based on this result, we select 6 SNPs within the region on chromosome 3 to design and develop the KASP marker. In this table, the 3 first SNPs were identified only in one population, but they explain a high percentage of the phenotypic variation, and we select them because of that. And the other 3 SNP, 4, 5, and 6, explain it a lower percentage of the phenotypic variation, but they were present in several populations. supporting their stability and robustness. So to validate the KASP markers, we use a set of samples from different origins that had previously been phenotyped for flowering. Here we saw the result of one of the KASP markers, and we can see a clear separation between the early allele and the late allele. So when we compare the phenotypic data with the KASP marker data, we found that the efficiency of this marker ranges between 81% and 19%, which is quite high for a quantitative trait like flowering time. So to develop KASP marker for Ascochyta blight, what did we do? So to develop those markers, we used 2 real populations, segregating for this trait, and these populations were phenotyped in the field over 2 years, and also genotyped using GBS. By association analysis, we identified 4 genomic regions associated with the resistance located on chromosome 2 and 4, and these results have been published in the study shown here, in case you are interested in more details. So to develop the KASP marker, we focus on the region on chromosome 4, since it was detected in both populations and explained the highest percentage of the phenotypic variation. In this table, we have the result of 2 KASP markers, AB1 and AB5, in the parents, and in yellow is the allele that carry the resistant parent, and in white the allele that carry the susceptible parent. To validate the usefulness of the KASP marker, we tested the markers in 90 genotypes that had been previously phenotyped, and in the graph, we see that the marker clearly discriminates the resistant allele from the susceptible allele. And when comparing the phenotype data with the data from the KASP marker, we saw that most individuals that were classified as resistant carry the resistant allele, and most individuals classified as susceptible carry the susceptible allele. Therefore, the efficiency of those markers were pretty high, 78% and 85%, which is very high also for a quantitative trait like Ascochyta blight. So what are we doing now, and what are our current objectives regarding Ascochyta blight resistance? As mentioned before, Ascochyta blight is a complex trait. So although we already have some useful markers, our current goal is to continue developing additional markers targeting other genomic regions associated with resistance. In this way, we aim to better capture the underlying genetic architecture of the trait and also improving the effectiveness of marker-assisted selection. We have already developed KASP marker for the region on chromosome 2. However, its efficiency is a little bit low. And therefore, we are now focusing our efforts on developing more robust KASP marker for the region on chromosome 2 and 4. To do this, we are resequencing 12 genotypes, 6 of them resistant and 6 of them susceptible, with diverse genetic backgrounds, in order to identify allelic variants that explain a higher percentage of the phenotypic variation. What about KASP marker for Fusarium wilt? How did we develop them? We built on previous work from our group, in which we use several real populations segregating for Fusarium wilt resistance. And through a fine mapping approach, we identify a 2 megabase genomic region containing 6 SNPs associated with resistance. Based on this SNP, we define the resistant haplotype and the susceptible haplotype for this region. From this set, 2 SNPs, SNP number 8 and SNP number 40, were selected for the development of KASP markers. The KASP markers were tested on a set of 90 genotypes that had been previously phenotyped as either resistant or susceptible. As Natalia mentioned before, having a well-characterized set of plant material with known phenotypes is essential for validating KASP marker. In this graph, we can see that the marker performed well, clearly separating the resistant and susceptible allele here, also resistant and susceptible allele here. When we compare the KASP genotyping result with the phenotypic data, we observe that most of the individuals classified as resistant carry the resistant allele, and most individuals classified as susceptible carry the susceptible allele. Therefore, the efficiency of this marker is also quite high, which confirms their usefulness for selecting fusarium-resistant material. How have we developed KASP markers for leaf type? In chickpea, there are two leaf types, simple and compound. And in our group, we have populations segregating for this trait, and we were interested in understanding its genetic control and identifying the genes underlying leaf type variation. The result of this work have been very recently published in the paper I mentioned here. And here also you can see a brief summary of the main finding. However, I don't want to go into the details as this is mainly focused on basic research, and this webinar is about the development of KASP marker. However, as you know, this basic research is essential in order to be able to develop robust molecular marker. So to develop the KASP markers, we resequence two genotypes with compound leaf and two genotypes with simple leaf. When analyzing the sequence result of these genotypes, we identify a deletion associated with the simple leaf type in the CANOX gene. And specifically, we observe the presence of a premature stop codon in the open reading frame in the simple leaf mutant. Based on this finding, we select the deletion to develop the KASP markers. We validate the KASP marker in a real population of 77 individuals. 34 of them were classified as simple leaf, 41 compound leaf, and 2 heterozygous. As shown in this graph, the simple leaf individual, highlighted in blue, are clearly separated from the compound leaf individuals highlighted in red. This result demonstrates that the marker has a very high efficiency of around 90% and is highly useful for selecting either simple or compound leaf types. So within the BELIS project, as Ignacio already mentioned, we are also carrying out a proof of concept studies in which we are applying the developed markers to a chickpea breeding program. This work would not be possible without the essential contribution of our partners, Cooperativa Campo de Tejada and Agrovegetal. Their close collaboration, long-standing experience, and a strong commitment to apply breeding have been fundamental for the success of this work. We have been working together for more than 20 years, and this partnership here you can see some of the material developed within the Agrovegetal breeding program. These are F2 plants derived from different crosses, which are currently being genotyped with the KASP marker. So far, around 1,000 F2 plants from different crosses have been grown by Agrovegetal, and these plants have been genotyped using the most efficient KASP marker for selecting ascochyta-resistant material, as well as the most efficient marker for fusarium resistant. In this table, we can see the result obtained with KASP marker for fusarium across 7 different F2 populations derived from different crosses. And the result fits well with the expected segregation ratio, taking into account that resistance to Fusarium race 5 in chickpea is controlled by one gene and is recessive. The marker has allowed us to select 264 F2 individuals carrying the resistant allele, which represent a major advantage for the breeding program because it reduces the number of individuals that need to be phenotyped in the field to confirm the resistance, and also will help to accelerate the breeding process. So in this graph, we can see the result of the KASP marker for Ascochyta resistant here and for Fusarium here. We tested the markers in 182 individuals from a cross in which both parents were resistant to Ascochyta, but differ in their response to Fusarium. As we expected for Ascochyta, all the individuals carry the allele associated with the resistance, and in the case of Fusarium, a lower number of individuals carry the allele associated with the resistance. We observe here that the segregation pattern fits well with the expected genetic ratio. So taken together, these results clearly demonstrate the reliability of the markers and their strong potential for implementation in breeding program. Also, they represent a powerful tool to accelerate the genetic improvement and to develop more resilient chickpea varieties with greater precision and efficiency. And with this, I would like to conclude my presentation, and we will be happy to take any question you may have.
Thank you very much, Patricia. Thank you very much, Natalia. Really interesting presentation. I'm sure that we may have some questions from the audience, which I would like to remind again about this Q&A section, question and answer section in Zoom for everyone to use. I actually saw that some people raised their hand in the audience, so please keep your questions. You can already type them in the Q&A section. We will use that option for you to ask questions after the next presentation and during the discussion. As mentioned, thank you both very, very much. We are now moving to our next contribution, which actually we have the contribution intervention from a guest speaker outside of BELIS. Today we are delighted to welcome Dr. Mariem Bouhadida. Sorry, Mariem. She's national focal point from the FAO International Treaty on Plant Genetic Resources for Food and Agriculture, also Associate Professor, Chickpea Breeder and Biotechnologist from the National Institute of Agricultural Research of Tunisia, INRAT. Mariem, thank you very much for being here today with us. I'm actually going to share my screen to put your slides. So let me put it like that. I guess that you are able to see that, correct?
Yeah.
So I let you to the floor is yours. Thank you very much, Mariem.
Thank you very much, Joaquin. For first, thank you for Siham Taragota for giving me this opportunity to participate in this webinar. It's a pleasure today to present you an overview regarding the chickpea breeding program at INRAT. And first, I would like just to present myself. I'm agronomist, engineer agronomist. Joaquin, if you can go to the next slide, please. I'm sorry, because I'm going to. So just a brief overview regarding my academic background. I'm an agronomist from INRAT, the National Institute of Agronomy of Tunis. And then I had my Master of Science in plant breeding from INRAT. Then I was in Zaragoza at the University of Saragossa doing my Master of Science and PhD on biochemistry and molecular biology. And then I'm at INRAT since 2008. I'm working as a researcher at INRAT. So INRAT, just a brief overview regarding INRAT. It was created since 1913. Next slide, please. it's created since 1913 and is the oldest agronomic research institute. And I have been working since 2008 in the field crop laboratory, dedicated to the breeding and improvement of cereals and legumes. So I'm coordinating the National Legume Breeding Program since 2018. Next slide, please. Next. To understand the key challenges of our activity, our research activity, they are closely related to the context of Tunisia, a North African country, highly affected by climate change, and this includes rainfall variability, rising temperature, soil degradation, water pollution, and increasing water scarcity. All these factors negatively impact the productivity and sustainability of cropping system and ultimately threaten food security. Next. Yeah, next, please. One of our challenges for our research activity are closely linked to the context of Tunisia. As I said before, and in this context, in the context of climate change, the Tunisian breeding program aims to contribute in the increase of the cropping area, improving the yields, next please, reducing the impact of climate change on legumes, and reducing the impact of diseases and pests on food legumes crops. Next slide. So in Tunisia, field crops cover more than 1.7 million of hectares, and they represent more than the half of the arable land. But unfortunately, legumes represent only 5% of this area, around 90,000 hectares, and chickpea represent unfortunately only 12 of this area. Next, please. So more than 85% of grain legume production areas are located in the northern region, where annual rainfall are superior than 400 millimeters. However, in the recent years, rainfall has become highly irregular. And in some seasons, this has significantly affected legume production. For next slide, in addition to the abiotic constraints, we are facing the biotic challenges causing significant damages. Mainly, for example, we have the problem of fusarium wilt, and also we have problem of ascochyta blight. So we have similar biotic constraints, like, for example, in Spain or the south of the Mediterranean countries. Next. We have also a big problem of Orobanche fetida, which is a hollow parasitic weed that continues to cause serious damages and is uncontrolled. These damages are really uncontrolled on legume crops, especially for faba bean. And as you can see here, for example, next slide, you can see here, for example, when we uproot the plant, we can see the Orobanche attack underground, so plant can be highly infested underground, and the only way to confirm that is uprooting faba bean, in this case, for example, faba bean. But this parasitic plant, unfortunately, is now attacking other legumes, next slide, like alfalfa, like common veg, narbon veg, and recently, this last years, we are observing the extent of the attacks on the chickpea fields, causing severe damages. So Orobanche fetida is becoming also a serious threat for chickpea. Next slide. So I would like to give a very brief overview of the 3 legume breeding program at INRAT. We have faba bean, the faba bean breeding program, the second one, the chickpea breeding program, and the lentil breeding program. All of them are focusing on creating or releasing new varieties with high yield potential, resistant to the major biotic and biotic stresses, good technological and nutritional quality, and the suitability for mechanical harvesting. Next slide. For that, we are conducting our trials in 3 main experimental stations. Go ahead, please. Yeah, we have 3 main experimental stations in Tunisia, belonging to different bioclimatic zones, where we are conducting our trials of faba bean, lentil, and chickpea, and where we are testing our material. Next slide. So the main objective of the chickpea breeding program, now we are going to focus more on the chickpea breeding program at INRAT. We are focusing on the release of or selection of lines with good resistance to Ascochyta blight, Fusarium wilt, growth habits, and having a large seed size, since it's the consumer have this kind of preference for the big seed size. We are also targeting to improve the yield, to increase the cropping area, and to improve the product marketing through a well-adapted value chain. Next. So the breeding sham start at the F3 generation, as you can see here. We received annually from ICARDA the material and the material that we received in general is a material F2, F3, so a segregated material, and it's evaluated in disease screening nurseries under field condition for resistance to Fusarium wilt and to Ascochyta blight. A pedigree selection method based on single plant selection is applied. Selected plants are advanced through progeny rows, where lines selected in the Ascochyta nursery are tested also in the following year in the Fusarium screening nursery, and vice versa. Once the F8 generation is reached, the candidate lines are entered into multi-location yield trials, and we identify the most agronomically superior genotypes, and the best performing lines are then proposed for release based on their resistance to disease, their agronomic performance, and their nutritional quality. Next slide. So as you can observe here, 100 of chickpea lines are tested each year in our experimental stations, just to have an idea about our trials in the field. Next slide. So in parallel with field conditions, lines are also tested under controlled conditions, particularly when we are targeting a specific pathogen or race. For example, for Fusarium, we have the race 0 of the Fusarium oxysporum C series, but sometimes we are also testing or trying to identify if there is other pathogens or other races. And also we do the same. So this is for Ascochyta blight, and we do also the same. Next slide, for Fusarium oxysporum C series. So we are conducting trials and experiments under controlled conditions, and we are following the scoring. For example, to Ascochyta, we are following in the field and under controlled conditions the radiancing disease, for example, rating scale. And for next slide, for Fusarium, we are following the Jiménez-Diaz and Trapero-Casas rating scale in order to identify the best lines that having the good scoring scale. Next slide. And what we discover, since we start observing symptoms of wilting on Tunisian varieties previously released for their resistance to Fokker 0, we start thinking about identifying causes of wilting symptoms observed. And we conducted several surveys in the main chickpea growing areas in order to do sampling and identify morphological characterization and molecular characterization in order to identify what's going on. So, apart the fork, for example, for Fusarium oxysporum C series race 0, there is other pathogen species involved in the wilting that we are observing in our growing areas of chickpeas. Next slide. So for that, also we used the sequencing of the elongation factor of our isolates or the strains that we prepared. So after our surveys, we did monosporal cultures for all our isolates, and we performed the sequencing, and also we use specific markers in order to identify if there is other thing apart the Fusarium oxysporum C series. The one who is tightly linked to wilting symptom in our fields. And we were able to identify a new species, which is Fusarium redolens, and we performed like pathogenicity tests in order to see if this new species is a pathogenic species. And after doing this pathogenicity test, we discovered that Fusarium redolens is also one of the species of Fusarium involved in the problem of wilting that we are observing in our chickpea growing areas. I think that here the problem that also we are now speaking about the complex of Fusarium, because it's not just limited to Fusarium oxysporum C series. There is other species involved in the Fusarium complex, which are a group of species tightly linked to problems of yellowing and wilting of chickpea. Next slide, please. I would like to speak a little bit about the use of molecular markers in the Tunisian chickpea breeding program. We used the Spanish, the result of the Spanish group identifying two markers tightly linked to two main QTLs directly involved in the resistance to Ascochyta blight. And we used the SCAR marker that was identified tightly linked to the QTL ER1, and another diagnostic marker, which is the CA-ETR, which is tightly linked to the second QTL. And we used these markers on our material. Next slide, please. And we were able to identify or to select basing on the, we did the selection on the fields, but we used this kind of markers, and we select the ones that have the best alleles, combination of alleles, in order to reinforce our breeding program, the conventional breeding program. And we were able to present two candidate varieties basing on the use of markers in our breeding program. Next slide, please. Now I'm going to present you the main achievement of that we did in our breeding program. Next slide. I'm going to start with the Noor, which is registered since 2011. This variety have been developed before, but I would like to mention that our main achievement from Noor, because it's the first variety to combine the dual disease resistance to Ascochyta blight and Fusarium wilt. And this is the first variety that combine both resistance, the dual resistance, with a large seed size that meets the expectation of the Tunisian consumer. Next slide. This variety was registered in 2015, and it shows a broader adaptation compared to Noor. Next slide. And this one, it has the particularity, it was registered in 2018, and it has the particularity that it shows resistance to the main viruses, the chickpea chlorotic stunt virus and the beet western yellow virus. Next slide. This is our last release. It was released in Zina, was released in 2023, and is distinguished from the previous one by a large seed size and improved nutritional and functional characteristics, making it better suited for processing with higher water and oil absorption. This aspect has become a key priority for us because leading to increased research efforts to ensure the marketability of farmers' production and stimulate market dynamic and support the development of structured chickpea value chain. Next, please. So as we may know, chickpea is a legume rich in protein, dietary fiber, and bioactive compounds, offering a strong potential for the development of innovative and nutrition food products. Varieties differ in their physiochemical, mineral, and techno-functional composition, which influences their suitability for incorporation into cereal-based products. Therefore, it's essential to identify the optimal variety to maximize both the nutritional and technological quality of the developed products. Next. So here I would like to show you the work that was conducted at the laboratory in collaboration with industrial partners and startups. It focused on developing a gluten-free product from chickpea flour and aquafaba. Example, we can see, for example, cakes, meringue, and also next, please. So the key aspect of this work was the selection of suitable chickpea varieties, since they differ in their techno-functional properties and functional behavior, which directly affect processing. So here you can see, for example, cereal bars, chickpea flour, and muffins based on chickpea flours and pomegranate syrup, chickpea flour, gluten-free biscuits. So we think that this is very important to think about the valorization of chickpea in the whole value chain. Next slide, please. So this is just to have an idea about the field days that we are organizing. So involving all stakeholders, farmer, extension agent, technicians, student, researcher, industrials, to present and demonstrate the technical package associated with each chickpea varieties released, and to disseminate the best agronomic practice and the real field condition. We're promoting the new developed chickpea and varieties, facilitating the knowledge and exchange feedback among all the stakeholders. Next slide, please. I would like to here to thank all our partners, colleagues, and among our partners, ICARDA is one of the previous partners with us in the breeding program, but also the University of Cordoba, the IFAPA. We were collaborating since, I think, 30 years ago, and we will be very happy to start to collaborate again in the future. And I would like, next slide, to thank you for your attention.
Thank you very much, Mariem. Thank you for this really valuable contribution to this BELIS webinar. We're really glad to have had you today, and we ask you to stay online with us in order to address any question that we may have after your contribution. Thank you once again.
Thank you.
So our next slide in the agenda is the presentation of the BELIS network by Dr. Claire Barber-Massen from the BELIS partner Sinovia, who is going to present the BELIS network, one of the really important part of the project. So Claire, I guess that you are here with us.
Yeah, yes, I'm here. Can you hear me? Yeah.
Loud and clear, yes.
Okay, great. So I will share my screen. Thanks a lot. I'm very sorry because my okay, I think I cannot. Maybe Joachim, could you share the screen for me?
Do I have your slides? Oh. No, but you can't share your screen, or? You should be able to.
Okay.
Let me check.
Wait. No. Okay. It's working now. Yeah.
Okay. Okay.
So I think. Okay. Can you see my presentation?
Yes.
Okay, great. So I'm very sorry because my camera doesn't work today, but at least you have my picture on this slide, so it's it's nice. So yeah, I'm Claire Barbet-Massin from Terrinovia. I'm involved in the BELIS project, in charge of the BELIS network that I will present you in a few words. So we have time for questions, and for the previous presentations that were of interest, and I've seen that some questions were already asked in the chat. So here is a few words about the BELIS network's aim. Indeed, in the BELIS project, we aim to deliver outputs useful for breeders, researchers, and all the value chain connected to legumes. And that's why we thought BELIS network would be the good gate to connect people in order, on one side, to make available the knowledge obtained during the project, and also to facilitate exchanges and collaboration among stakeholders. So here there is this green sentence. The network will be a gate to transfer BELIS output and also to connect legume breeding actors across EU and associated countries. So based on these 2 points, can I move? So first of all, BELIS network activities. So when you become a member to the BELIS network, you will have a facilitated access to BELIS results through the BELIS newsletter, which will be shared to you. Also, some surveys can be shared to identify needs for webinar or for trainings, thematic of interest, etc. And some webinars can be organized either to present some key project outputs or on specific topics such as chickpea today. We can also organize some session focused on stakeholders if some stakeholders are willing to be more visible, to present their work and also to exchange one with another. And also in the BELIS project, there is a training activities session. So there will be some training session organized by SIAM. It will be in-person session, and to be a member of the network will give you the opportunity to be informed about these training activities and eventually to register if you are interested too. And the other part of this BELIS network is to facilitate exchanges and collaboration through in-person meeting. And for instance, I put an event that is coming soon because I put the 8th of January, but it's of June. Sorry for the mistake. I will correct it after this webinar. So in approximately 1 month, there will be this ELS side event at Dubrovnik. It will be a workshop on the Monday afternoon before the ILS conference starts. And the aim is to present 4 European projects, among which BELIS, and also the Increase project, the Legume Generation project, and the Prosper project, and to present outputs from these different projects. So you can see here an idea of in-person meetings where people will meet, exchange, and among projects, and it can be an opportunity to create new collaboration. Also, some online meetings can be organized, as I said before, so with short presentations from different stakeholders, round tables, etc., for people to connect. And also, if some stakeholders are interested in connecting and working together, the BELIS network will be happy to help study funding opportunities from national or international calls. Yeah, here it is. So why to join this BELIS network? It will give you a very easy access to BELIS outputs, the opportunity to collaborate with experts, to participate in events, and also to become a member of the BELIS network is free until the end of the project, September 30, 2028. And now how to join. So if you go on the BELIS website, I will put the link in the conversation after my presentation. You will find in the About section BELIS network link. And when you click on this BELIS network link, you can join the BELIS network through an online membership form. And yeah, that's quite easy to fill. And here it is. Thank you very much for listening to me.
Thank you, Claire. Thank you very much. As usual, I have put the link to the to join in the newsletter as well as the network. I already put it in the chat for everyone. And also for everyone to know that all the presentation of this webinar will be available through the BELIS website in a post that we will be publishing after the performing of this session. So thank you all very much. According to the schedule, now we have some time for questions and answers. Especially we expect some questions from the audience, but actually we already started to have a discussion in the chat. And I don't know if some of you would like to complement something about what you reflected in this brief discussion, maybe Bernadette or Patricia and Natalia, about this availability of the use of these markers for researching groups.
Yes, the markers. Can you hear me?
Yes.
Okay. So the markers for, as Natalia already answered in the chat, the markers for flowering time and leaf type are already available. They are already published with all the conditions that you need for the master mix and everything. So the ones for Ascochyta blight and Fusarium wilt, we haven't published yet, but we hope we will publish soon. If not, maybe we can make them available for the BELIS project researcher. If they are interested, maybe we can make that available, but maybe I have to talk about this with Ignacio and the other partner.
Thank you, Patricia. Something that anyone would like to add into this, or we can move to any other topic? From the audience, please, as mentioned, you don't hesitate to use the Q&A section for questions or the chat, as you have done already. Yeah, it seems we have, apart from thanking your your explanation and sharing, yeah, okay. So I think that can take me to some other questions that we have that I would like to raise to Patricia and Natalia, according to the presentation. The main backbone of the presentation of this webinar today is about the which stage of the breeding process would be kind of benefit the most from these markers. Could be early generation screening, line advancement, or parent selection. Anything that you would like to comment of that?
I think it's better use it in the early generation, the breeding program, because Patricia explained before, you eliminated a lot of material or individual that are not with the allele or the phenotype decided. But in the other case, parental line or the advanced lines, it's good you see because you can characterize your material. I think it's used if you have a KASP marker is useful in all stage of a breeding program.
Thank you, Natalia. There's another question that I would like to ask to Ignacio in this case about the breeding priorities in the under Mediterranean conditions. Can you explain us how these breeding priorities has changed in the recent years in the basin of the Mediterranean area?
For us, the most important characters to be improved are related with quality, because a few years ago, the chickpea was a commodity, staple crop, but now we are trying to change it to a high quality product, because the international prices for pulse crops are very, very low, are 30, 40 times a kilogram. And with the international price for the normal chickpea, the farmers in Spain don't have profits. Then we need to work with protected geographical indications to make one type of chickpea that is paid twice the international prices to be attractive for farmers, for seed company, for everyone. Then in the south of Spain, we have one type of chickpea, the blanco lechoso, that is not found in the international commerce. And then people likes to eat these very big and white seeds, and they pay twice. And our breeding is linked to this quality. And we get profits as seed producers because people who want to produce the high quality, high price seeds, they pay for certified seed. For the common small seeds, the people reuse the seeds. Then we are a private company and we put a lot of focus in the quality. But we are not interested that all the market was this type of seed, because if everybody produce blanco lechoso, the price will decrease. But we want to conserve this 20-30% of the market of high quality with high prices in parallel with the normal one.
Thank you, Ignacio. Thank you for that. So we have some questions in the Q&A section, and Natalia already addressed some of the questions from Dr. Mahmoud Said. Thank you, Natalia, for that. But we have a pending question coming from Losari Navi. Sorry if I don't pronounce that correctly. The question is about how were seed quality traits studied, seed size, shape, and color. What was the methodology? And who would like to take this question?
We do it visually. We have no markers, no technology, only to make crosses with the traditional blanco lechoso type. All the other characters are improved with markers and so on. But for size and color, we do visually.
Thank you, Ignacio. Yeah, there's another question in the chat, no in the Q&A, but in the chat from Dr. Priti Chakhar. It's about related of the KASP markers, so I guess this is for Patricia and or Natalia. The question is about, can this marker develop for disease resistance in megazonas, disease development affected by host, pathogen, and highly variable environment testing conditions? Patricia, you are muted.
Yeah.
Yeah.
I was answering in the chat.
Thank you.
But it's okay. I think I'm not sure if I truly understand the question, because in fact, with the KASP marker, you are selecting by the genotype, so it shouldn't be affected by the environment, because you are selecting by the genotype, but it is true that if there are different races or different pathotypes of the diseases, you need to have well characterized all the phenotype, because the genes for each race or pathotype can be different. So you need to develop KASP marker for each specific region of the genome that is related with the specific pathogen, pathotype or race or gene, if there are more than one gene affecting the disease. So I'm not sure if that's the question or.
Thank you, Natalia. I think so, but Dr. Priti Jakhar, in case you would like to re-ask or something, please don't hesitate. I see some thumbs up in the reaction, so it seems to be good. Yeah, Natalia.
No, I agree with Patricia. It's very complex, the question. I think you have a clear or a region genomic very highlight, because the KASP is associated with the trait.
Mm-hmm. Thank you, Natalia. There's a question that I would like to address to Mariem about your intervention today. Thank you for presenting the status and challenge and prospect in Tunisia. And regarding to that, I would like to ask you about, maybe it's an obvious question, but about the local varieties. And I would like to ask you to please explain how important are local varieties and farmer preferred traits in Tunisian breeding strategies.
Well, thank you. Thank you, Joaquin, for this question. Local varieties and farmer preferred traits are central to Tunisian breeding strategies. They represent a key source of genetic diversity and are well adapted to local agroecological conditions. And farmers' preferences, such as yield stability or grain size, for example, or cooking quality, and there is also the market acceptance. These things are very essential to ensure adoption of new varieties. And in our breeding programs, we are integrating the participatory approaches with farmers. This is to ensure that the released varieties meet the agronomic performances and also meet the needs of farmers. And even we are thinking about also the industrials, because we are, okay, we can meet the needs of farmers, but we are thinking about the valorization also of the chickpeas. So we are trying to adopt as a participatory approach where all the stakeholders are involved, farmers and industrials and also consumers. So this is our how to say, our strategy but the local varieties are involved in the crossing, and as you know, Tunisia isn't a center of diversity for chickpea. So we are mainly doing breeding. So the local varieties are varieties performed or developed in our breeding programs. So we don't have, for example, the autochthonous varieties for chickpea, but yeah, the program released chickpea varieties since 1997, the beginning of the 2000s. So yeah, we have about maybe 20 varieties of chickpea, local variety, but released by the program.
Thank you, Mariem. I think that's definitely really important, and also the collaboration that you have with a partner like Icarda, which is actually part of BELIS as well, is crucial in all that sense. Thank you for that. There's a question that I would like to ask to Patricia and Natalia, and you've been presenting some different traits in chickpea. And do you think that are there any other key agronomic traits that should be that shall now be target for future KASP marker developments? Something that, sorry, I think that Ignacio, you raise your hand and maybe you would like to point out something before we address that question.
Okay, thank you. It's a question for Mariem. I am interested if they have breeding for chickpea for feed or only for food. In Tunisia, do you use the chickpea for feed or animal feed?
No, we are releasing varieties only for food, not for feed. Faba bean, we are using faba bean, the minor type for feed, but not for chickpea. Chickpea is essentially for food.
Okay, I have done this question because in our trials in Andalusia during a lot of years, we see that the farmers also use faba bean and pea for feed. But in our trial, the average yield of some small chickpeas is even higher than faba bean or pea. I think that we can reuse the chickpea for feed because long time ago, 60 years ago in Spain, the chickpea was also grown for feed. We have lost this use, but we think that may be interesting because in some cases, some cultivars are very high yielding, even more than faba bean and pea in our conditions. In our condition, the objective for yield is around 2 tons hectare, and we have this yield or more with some small chickpeas.
Yeah, maybe we need to think about that. But since the most growing area in Tunisia, it's dedicated for faba bean, the minor type. So I don't know if chickpea will have his place because we need to think about it.
Okay, thank you.
Thank you for.
Yeah, thank you both for this discussion, and please don't hesitate to keep on contributing to that, to the discussion. We are getting to the last minutes of the webinar, but there's still this question that I would like to come back to it to be addressed to Patricia and Natalia, as I was mentioning about this. Thinking on the future of KASP marker development, are there any other key agronomic traits that you would like to point out that could be target for this future development?
Yes, well, right now we are working on growth habit, and there is a PhD student from our group that is working on that trait. And we have also several populations segregating for the trait, and we have already identified the genomic regions and the genes that are involved in that trait. In the next step that we are going to do is develop the KASP marker for this growth habit, because in chickpea we have the arid, semi-arid, and the post-trait, which is coming from the wild. So we are trying to develop KASP marker for arid and semi-arid habit.
Thank you, Patricia.
Yeah.
There's actually a question by Bernadette in the chat about the. I think this can be an interesting discussion about the if the number of seeds or size of pod can be a yield component. Yeah, Bernadette, would you like to?
Yes. I don't know very well chickpea, but so I'm wondering if there is a single seed in each pod, or maybe sometimes 2 or 3, and if the seed size is related to this number of seeds per pod.
Yes.
And if ever you can increase the number of seeds per pod, would you achieve a better seed yield per surface unit?
From my experience, the main part of cultivars, at least in the big size cultivars, are always two seeds per pod, and sometimes you have 1 seed per pod, but the influence in low yield is very related with very low yield. Then, if you want to have a good yield, you need two seeds per pod. I don't know if there is cultivar with 3 or more, but in my conditions, all the commercial cultivars are 2 seed per pots. There is cultivar with tendency, we have only 1 seed, but normally these cultivars are discarded because very low yielding. Even with high seed, but low yielding, very low yielding.
Thank you, Ignacio. Any comment on reaction on that?
Only that there are some genotypes that they have a double pot, only that they have only 1 seed in each pot. We have also some molecular studies about that trait, and we identify the gene that is related to that trait. But we didn't develop KASP marker for that, so maybe could be also another targeting region for developing KASP marker. So.
And would it be interesting to look for varieties with 3 seeds per pot?
3 seeds per pot?
Yes, to increase the yield.
I'm not sure about that. The only thing that we have seen that the lines that have double pot, it seems like they are more stable. The yield is more stable, more uniform in different environments. But in our fields, in our work, we've never seen an increase in the yield with this double pot. So maybe it's something to keep in mind and try to study. We don't have any data about that yet. So.
Okay, thanks.
Thank you, thank you both, and thank you all, because I think that we are running out of time and we should close the session. It's a pity, because I think that I have the feeling that we could have a discussion for much more minutes during today, but we should need to stick to the plan and to the time allocated for this webinar. So before closing, I would like to thank all of our speakers today. Thank you, Bernadette. Thank you, Ignacio. Thank you, Natalia, Patricia, Mariem, and Claire. Thank you very much for your valuable contribution. And thank you very much to everyone connected online. As mentioned, please keep an eye on BELIS website. We will be posting the video of the webinar, the slides and materials, and you already have available information about the project. To subscribe to the newsletter or to be part of the network, all the information and links are in the chat. And well, that's it. This is the end of this webinar. KASP marker linked to key agronomic traits for chickpea breeding, organized in the framework of the Horizon Europe Project BELIS. Thank you to all who joined the session and our speakers today. And we're looking very much forward to keep in touch with everyone. We now close this webinar. Thank you very much, and take care.
Thank you.
Bye. Thank you.
Thank you.
Bye-bye.
Bye-bye. Thank you.
Bye.